Andrei Gudkov asked me to explain the image. It's a painting from 1546 by Cranach the Elder, and some of you will notice a directional movement of the infirm and elderly through a magic elixir to restore life, youthfulness, and longevity. The system that I'd like to start with in this session has to do with the proteome. And I think something that I'd like all of you to recognize is for every process in the cell, for every polymer, the synthesis of DNA, the transcription of all genes, the making of all proteins, how cells achieve their shape and their form and maintain it throughout their lifespan is dependent on proteins. And one thing that we can appreciate is quality control as perfect as we age as it was when we began? For every polypeptide, it starts off as an ensemble of intermediates. They form off-pathway oligomers and aggregates, which, as you know, is a major problem in all biological systems as they age, whether it's an in vitro test tube reaction all the way to any cell within a body. Molecular chaperones, the proteasome and autophagy, play the major mechanisms for clearance and preventing misfolding, thus preventing off-pathway intermediates. Molecular chaperones, of course, are the major proteins that guide the stability of the proteome. An important thing to consider is how do we achieve proteome quality control given polymorphisms and mutations, recognizing that at least half of the proteome for each of us has a missense mutation. Then if you add to it biosynthetic error, which is significant, 10 to the minus third to 10 to the minus fourth for every biological step, of which there are dozens of steps between the initiation of transcription and the folding of a protein. Thus, biosynthetic error contributes up to nearly 20% of proteins made have mistakes. Then, of course, all of you are familiar with energetic deficits, changes in energy metabolism. All of this, of course, leads to protein damage, and aging, of course, as I think has already been pointed out, is probably the major trauma to biological systems. Proteostasis then represents this balance between function and dysfunction. So this idea behind proteostasis is literally a balance. And it balances the levels of molecular chaperones, proteasome autophagy, with these mutations and these biosynthetic errors, which lead to damaged proteins. The problem, of course, is that the system is not static, and it changes dramatically. One change, of course, is aging. Once aging occurs, it pushes a little bit harder on the balance. It has a more difficult time. That then, of course, if you add to it proteins like alpha-synuclein, Huntington, tau, A-beta, you now have a situation where the system struggles to restore balance. What I'll tell you a little bit about as we go towards the end of the talk is how we've identified small molecule proteostasis regulators that can restore the balance to the system, and in doing so, affect protein misfolding. Now, the proteostasis network is literally defined then by a series of upstream genetic switches. They include, for example, the heat shock response, which regulates all chaperones, the unfolded protein response that regulates chaperone clearance systems in the endoplasmic reticulum, FOXO that you've already heard about at this lecture, which of course is a metabolic stress response, the proteasome, calcium signaling, oxidative stress response. Each of these then ensure that for every polypeptide that a critical triage decision's made. Instead of the off-pathway aggregation occurring, these proteostasis networks ensure that the system is driven towards clearance or folding. thus maintaining folding equilibrium. Now, the composition of the proteostasis network at this point is nearly 1,500 genes, corresponding to 300 molecular chaperones, the E1s, E2s, and the E3s of the ubiquitin proteasome system, autophagy. If one looks then at the expression of these, shown at the, so this is now a transcriptome analysis where you're looking at the expression of different members of the proteostasis network and organized according to tissues where red is abundant and green is repressed. What you can see is that there's a very big tissue difference. For example, the expression of chaperones, proteasome clearance apparatus in the brain is strikingly different than, for example, in the immune system. It is very important to recognize that even though all proteins have the same fundamentals of helices and sheets, that the relative proportion of those that are in each compartment or secreted varies dramatically according to tissue. In other words, the proteostasis network is highly tissue specific. If one then adds to that the effect of aging, what I'm showing you now is a transcriptome analysis of the human striatum. What you're looking at in red are the genes that are expressed in individuals who are young, and you can see that the same set of genes are actually repressed as humans get old. So this is from the striatum. If you look at the average age distribution, you can see that the genes are abundantly expressed in individuals that are about up to 50 years old. Once you hit the age of about 65, the same genes are shut down. So you might say, well, what genes are these? These are the genes that encode your chaperones. These are the genes that encode the components of the proteasome. So the major quality control system changes significantly. Now, what's intriguing in this data set is that there are interesting outliers in this cluster analysis. For example, there are 3 individuals who are age 70, 82, and 75 whose proteostasis network maps to a much younger group. Now, that's interesting and provocative. If one did a longitudinal study, obviously you can't in this kind. These are human brain striatal samples. But if one could do a longitudinal study, are these individuals protected against protein misfolding diseases? Are they protected against Alzheimer's, Parkinson's, ALS, and all the misfolding diseases? Likewise, it's equally interesting that there are 2 individuals who are 36 and 44 years old whose proteostasis network transcriptome places them into a much older group. A similar question can be posed. Are these individuals much higher at risk? In a sense, their proteostasis network allows proteins to be misfolded. So some of the questions for the future is, does the PN age at the same time in different tissues at the same rate? And what happens in age-associated neurodegenerative disease? What I'm showing you now is just the molecular chaperones. So this is organized according to the 300 molecular chaperones. It's a combination of protein-protein interaction and transcriptome. Red is that you're induced. Green means you're repressed in human aging. And I think you can see that there's 3 clusters that are induced. The majority of molecular chaperones are repressed. So you have to consider this, that the actual repression that we see, at least by a systems network analysis, could this, in a very simple way, provide the explanation for the accumulation of misfolded species, aggregation, and a general reduction in the robustness of cellular systems. So this gives you an idea of how we've created a proteostasis network with different subcomponents for autophagy or the proteasome. What we're moving towards is now a specification to specific tissues. Not just neurons, but neurons in the context of tissues, neurons in the context of an actual animal. And obviously the model system that I'm showing you is that of C. elegans. It has to be done, I think you've already heard earlier from Robert, one of the values of C. elegans, you're going to hear a lot of other talks about C. elegans, but the advantage is an organism for which lineage is precisely known and all the genetic tools are available. So one then asks the question, how is proteostasis regulated in a metazoan? I want to remind you that in this animal, the heat shock response, which is there to protect the proteome in every cell and tissue, is actually regulated by two sensory neurons all the way in the head. That's how the animal senses environmental stress, and it transmits the information all throughout the soma. This ensures that the tissue response to stress is coordinated to promote organismal survival. Another theme that came out of this morning's discussion, that we have to think about the protective mechanisms that enhance survivability, not as much as what causes pathology. We can then ask questions like, is proteostasis compromised during aging? The results that I showed you by systems analysis of human transcriptome data are daunting, but it's not experimental data. What happens now when we look, for example, in C. elegans, and can we re-engineer the organism to restore proteome health? So I'm going to describe two types of folding sensors. The first class is actually a metastable protein. Metastable proteins harbor missense mutations, so at the permissive temperature, they fold perfectly fine. At the restrictive temperature, they don't fold well at all. This, by the way, is the fundamental of almost all of genetics and development, the use of TS mutations to map genetic pathways. We're going to use it as a folding sensor. It's well established that at the restrictive temperature, a single misfold leads to a loss of function. In a mutant phenotype, chaperones suppress this and enhance the folding to a functional state at the permissive temperature. It's important to keep the thermodynamic. That 10 degrees difference is a thermodynamic, a physical biochemistry of protein folding. So you can see, for example, with a temperature-sensitive mutation in paramycin, at the permissive temperature, 15 degrees, the animals move perfectly fine. There's no uncoordination, and the muscle striation is perfect. As the animals are raised at increasing temperature to 25, now you can see the muscle is destroyed and the animals stop moving. So you can monitor individual proteins and phenotypes. The other is fluorescent reporters. So these are fluorescent reporters such as luciferase, which itself is a metastable protein in a tissue of an animal. In this case, it's tagged with YFP, or what I'll describe today is the use of polyglutamine, which, as you know, is the basis of Huntington's disease and 9 other CAG repeat diseases, as a folding sensor. So let me describe an experiment. It tests the hypothesis of when during aging does misfolding occur. We're going to use a TS mutation in paramycin. If you look at a wild-type animal by immunostaining for paramycin, where you're looking at muscle striation, you can all notice that the muscle looks perfectly fine. When the animals are 2 weeks old, you can now start to see some disruption in fibrillar structure. For the temperature-sensitive mutation shown below, you can see at day 1 it's indistinguishable from the wild type. By day 3 and beyond, the paramycin has massively misfolded, and the consequence is that you can see that 50% of the animals have disrupted muscle cells by day 4 of adulthood. So this asks actually and answers an interesting question. Misfolding occurs, and it occurs not mid or late in adulthood, but early in the adulthood of the animal. So we can ask lots of questions about when this occurs and how about other tissues. And I'll just quickly summarize. RAS, which is a protein very important in signaling. A point mutation in RAS results in 50% of the RAS misfolded at day 6. Acetylcholine point mutation expressed in neurons. Day 4, GAS1, a mitochondrial matrix protein, 50% loss at day 4. And for perlecan, a muscle protein, 50% loss at day 5. So this answers something intriguing. It says, misfolding in C. elegans happens in animals as young adults. We don't have to worry about being middle-aged or old-aged. It's happened. Now the question is, why? So we decided to look at the cytoprotective stress responses, which made, I think, a logical next step. We looked at the heat shock response and the unfolded protein response. As you know, the heat shock response protects all proteins in the cytoplasm and the nucleus. The unfolded protein response protects everything that goes through the lumen and is secreted. What you're looking at is the inducibility of 4 different heat shock genes, 2 HSP70s, 2 small heat shock genes. We normalize in day 1 young adults to 100% full inducibility. These are synchronized animals. If we look at the population 3 days later, I think you can see that all 4 heat shock genes are not induced to the same level. They're reduced by a factor of 3 to 5-fold. And it remains non-inducible all the way through. Likewise, for the UPR, it's fully inducible at day 1, as is the heat shock response. By day 4, it's only 50% inducible, and by day 7, the UPR is not inducible. So this is intriguing. At this point, we do not know cause or effect. But if you're missing the heat shock response and the unfolded protein response, the organism can't respond to any misfolding stress. It undoubtedly provides an explanation for why misfolding occurs in young adults. Now, we did one more experiment. We asked then, could we re-engineer the organism? Re-engineering is, given this reality that misfolding occurs in young adults, could we activate, for example, chaperones? Could we activate, for example, FOXO3, which has been already described today? FOXO, of course, has been very well studied in C. elegans. It's DAF16, which is the major regulatory pathway downstream of DAF2 in age 1. So here's the misfolding of dynamin, 50% loss of folding at day 2. If, however, we activate HSF or DAF16 in early development, we now get protection all the way out to day 10 to day 12. If you're a worm, this is remarkable. It's not subtle at all. We've literally taken a protein that misfolds on day 2 of adulthood, and now it misfolds at day 10. There's a little trick here, and this also, I think, reflects a comment made earlier. In order to get this protection, we have to reset the proteostasis network during development. If we set it in adulthood, it's already too late. But that makes sense because you already can see the misfolding occurring in early adulthood. Now, these 2 pathways are not chosen by accident. Our laboratory and Cynthia Kenyon's had already demonstrated the critical importance of DAF16, age 1, and DAF2, and that heat shock factor was essential. So in other words, if you're missing the heat shock response, the other lifespan pathways don't work anyways, because you have to protect the proteome. In other words, regulation of proteostasis, cellular health span, and organismal lifespan require these 2 genetic pathways as one of the essential upstream pathways. What we like about this is this idea of youthful proteostasis, that by enhancing this system, we can shift misfolding to a later state. Now, the other folding sensor that we've used extensively are those associated with misfolding diseases. One that I'm showing you here is Huntington's disease, one of 10 polyglutamine diseases. And as I think all of you are familiar with, that you have a dramatic age of onset as the CAG repeat expands beyond 35 to 40 residues. We've made a number of C. elegans models that are shown here. As the length of polyglutamine goes between 35 and 40, you go from soluble to aggregated. This you can see as the animals age. So if you take Q35 animals, as young adults, there's no aggregation. As the animals now become day 4, day 5, you can see the profound aggregation that occurs. With this aggregation, you get a dramatic loss of motility, in other words, toxicity. So shown in red is the result for Q35. As the aggregates appear at day 4 and day 5, shown right here, you can see all of a sudden we go from no toxicity to now the animals show a loss of movement. Now we can use this for a variety of different approaches. What I want to share with you is how we use this to discover the genetics of proteostasis. This idea behind a balance where there's proteins that push on the system as they become damaged, the role then of these obvious protective mechanisms, the chaperones, the clearance machinery. But we use genetics in the same way that you heard earlier from Robert and others to discover new things. So what else is there? I've shown you that the animals show this very striking length-dependent aggregation that makes it a powerful visual tool for genome-wide screening. Moreover, it shows striking age dependence. So we can use this as a way to discover genes that enhance aggregation or suppress aggregation. So I'm just going to show you a couple of results. It I think, points out how relatively straightforward this is. If we take the Q35 animals, we start out with 17,000 genes. This is Robert probably didn't describe this earlier. In worms, it's particularly easy to knock down every gene because they eat bacteria, in this case engineered with double-strand RNA. And so using robotics, it's easy within a matter of 4 weeks to do entire genome-wide screens and phenotypic screens for anything, for aggregation, for movement, or any other phenotype. So out of the original 17,000 genes, we discover 155 genes that suppress Q35 aggregation. Of these, 88 suppress another polyglutamine that's longer. And of those 88, 64 suppress a completely different protein called SOD1 G93A, which is one of the mutations in human SOD1 that leads to ALS. So here's the first interesting result. That screening for suppressors of polyglutamine identifies suppressors for a completely different aggregation-prone protein. It means by genetics, they overlap. Now we can ask the question, how many of these, by suppressing aggregation, also suppress toxicity? So if there's a one-to-one correspondence between aggregation and toxicity, the prediction is all 66 should suppress toxicity. But instead, what we find, it's only 23, 1/3. 1/3 of them have no effect, and 1/3 of them suppress aggregation and increase toxicity, which tells us something very important, that aggregation and toxicity are genetically unlinked traits. We then ask, since these are all transgenic lines, how many of this will restore general proteostasis by restoring the folding of temperature-sensitive mutations? And we discover only 9 of these 23 now restore temperature-sensitive mutants. In other words, at the permissive temperature, where I've shown you aging leads to the aggregation, now we can prevent the age-dependent aggregation and misfolding. This gives you an example of how this looks. Here's the aggregation. These are just different animals. So you can see the aggregates, the suppression's quite dramatic. Here's the restoration of motility. Here's suppressors that increase the toxicity. Here's suppressors that are essentially neutral. And shown down here are the suppressors of G93A, SOD1. I think you can all see the aggregates. The protein's now soluble. I want to just summarize and tell you the result of one interesting new gene. And this brings in a completely new pathway. And you can see how dramatic this suppressor is. So here's the full-length animal Q35. Here it is when we knock down the gene GEI11. And I think you can see at increasing magnification that indeed there are no aggregates anymore, visually, biochemically, or biophysically. And we know that this suppression of aggregation is absolutely dependent on the adduction of cytoplasmic chaperones and absolutely dependent on activation of heat shock transcription factor 1. Now, GEI11, however, is a very interesting gene. It regulates the acetylcholine receptor. So let me just remind you what I'm telling you, that reducing the expression of GEI11 activates the chaperones, leading to suppression of aggregation. So I'm just going to summarize what we've learned. GEI11 is a MIB DNA binding protein conserved to humans that regulates the acetylcholine receptor, the L-type acetylcholine receptor. There's an increase in the acetylcholine receptor when there's a burst of acetylcholine going across the synaptic junction. So this is how, at the synaptic junction, you maintain balance. That increase in the L-type acetylcholine receptor leads to then a activation of the calcium channel, AGO19. That releases calcium to the ryanodine receptor, which then leads to contraction of the muscle and simultaneously activates the calcium-dependent kinases, calmodulin, which then phosphorylates heat shock factor, leading to its activation, movement into the nucleus, activation of chaperones, and protein folding. Why does an organism do this? What is contraction? Contraction is a muscle machine that has to be utilized very effectively. The activation of chaperones is a protective mechanism that ensures that the muscle mass is effectively utilized. So what I've told you about is this idea of a cellular challenge and cellular toxicity that results from decreased chaperone activity and protein homeostasis during aging and chronic stress. What I want to end with is just a few slides to give you a sense of therapeutic strategies to restore proteostasis. So what I'm showing you here is a cartoon. This is how heat shock factor is regulated. In all cells of all organisms, there's HSF1. It's inert, negatively regulated by chaperones. Upon stress, it trimerizes, binds to DNA. HSF then induces the transcription of heat shock proteins and chaperones. It's regulated by SIRT1. So this now becomes very intriguing. SIRT1 is bound to the human HSP70 promoter constitutively, and it keeps HSF in a DNA-bound state. HSF is also acetylated by CBP300, and when it does, it releases from DNA. So this is a very critical step in maintaining chaperones bound in the active state. So we generated cell lines with a reporter of the human HSP70 reporter. We screened 1 million molecules. We identified some nearly 400 that fell into 19 different chemical classes. I'm just showing you some of the chemical classes. Almost all of these molecules represent new chemical matter, even though there have been other small molecules that we and others had identified. Every one of these compounds activated HSP70 mRNA. They all elevated the different cytoplasmic chaperones by Western blot. By gel shift assay, they activated HSF DNA binding, and by chromatin immunoprecipitation assay, the HSF was bound to the endogenous HSP70 promoter as predicted. Most interesting is that some of these compounds are highly specific. Here's an example of a compound D1 that only induces HSP70, another compound F1, which is a barbiturate-like compound that activates equally well HSP70 and the oxidative stress response, shown here by HO1. When we test these compounds, I'll just quickly describe this. These are PC12 cells expressing Huntington. In the presence of the different molecules, the aggregates are gone. You can see that the aggregate count is disappeared. This is in CFTR. This is restoration of chloride channel. As you'll know, CF is a mutation delta F508 in the cytoplasmic domain, so it requires cytoplasmic chaperones. And when we use these compounds, it restores folding. Finally, the compounds even work in C. elegans. Here's the Q35 polyglutamine, and here they are on different small molecules. Here's the suppression of aggregation, which is quite dramatic. Here's the restoration of motility, and in every case, they activate only cytoplasmic chaperones. So this really gives us a sense, then, that it is possible to target this system, to upregulate it, and in this case, to restore proteostasis. So what I've told you about in my talk is this importance of the proteostasis network, that under normal conditions, it's what maintains the balance. It allows our polymorphisms not to be expressed as mutant proteins. However, aging represents, we think, the major stress. It causes other disease-associated mutations, such as Huntington's, such as A-beta, such as alpha-synuclein, to misfold. The combination of these events then competes for the limiting proteostasis network, which then leads to misfolding of normal cellular proteins, what we call collateral damage. So just to thank the people in my laboratory who did the work I had a chance to describe to you. Barbara Calamini led the group for the small molecule screening. Vina Prahlad did the work on the neurosensory system. Tali Gadalovitz and Janine Kirstein did the work on sensors. And Katarina Silva described the work on the GEI11 modifiers. And with that, thank you very much. Andre? Andre?
About the latest part with the small molecules. Did I understand you right that what they do, they simply create, they precondition a type of a heat shock response and make the system pre-prepared to the stresses, and the heat shock itself, if you do it mildly in the beginning, would do the same?
Actually, they're not equivalent. A heat shock itself is responding to the flux of misfolded proteins induced by that stress. The use of the small molecules activates the chaperone system and other cytoprotective mechanisms. So in some cases, they're activating SKIN1 or NRF2. Some cases, they're also activating the UPR. What they're doing is they're setting up a proteoprotective environment. So we think it's establishing an environment so that when you hit the cell now with a chronic aggregation-prone protein, the chaperone system has a little bit more capacity.
The other question is about one of the earlier slides, in which you mentioned that in the C. elegans, the pre-adaptive stress response is regulated by two neurons. And what does neurons actually do? Do they change the ability of the heat shock response to be regulatable?
So these are called the AFD thermal sensory neurons. When they are genetically ablated, there is no heat shock response in any tissue of the animal. Even though HSF and the chaperone system is still functional, it's lost its ability to respond. So we know that if we now take these AFD deficient animals and expose them to a different type of stress, like heavy metals or oxidants, now the heat shock response is absolutely fine. But if you now shut down the neurosensory system for oxidative stress or metals, now there's no activation by oxidative stress or, in some cases, for metals. So what this is telling us is that of the 302 neurons, that a subset of them, the sensory neurons, are actually designed to sense specific environmental stress signals, and they transduce this information to tell the soma which stress are you responding to, and to make sure it's orchestrated.
But since HSF1 is simply released because there's a competition for the chaperones, how can you influence the system by neurons?
We don't know the answer to that. We know that the intermediate is through neuropeptides and neurohormones, which must be setting up and affecting, I think, a new regulatory pathway that prevents the release of HSF from the chaperone system. Yes?
So some years back it was reported, I think, from Tom Johnson's lab that early induction of the heat shock response would be a predictor of longevity in otherwise isogenic single populations of worms. Would that mean that these worms actually have an enhanced, augmented proteostasis network? And what would be the cause of induction of this heat shock response so early in their life?
Yeah, the results that you described from Tom, I don't think he or anyone knows yet what's the basis of that, why in a population there's a small group that has a more robust heat shock system. And actually, it's even an assumption, right, that it's a more robust heat shock system. But there's something about those worms, as monitored by HSP16, that suggests that a better induction of HSP16 leads to better lifespan. But you know, HSP16 is interesting because it's also one of the few chaperones that's co-regulated by DAF16 and HSF, whereas HSP70 is much more HSF specific. So I think this is probably monitoring both metabolic stress and environmental stress at the same time. That's why it's so effective.
It's like the findings with the human populations where you have a proteomics and a proteostasis network that is induced or more robust in elderly, some elderly people versus some other young people. So could be that stochastic reasons.
Yes. Oh, look, obviously I fully believe so. That the link between the human systems network analysis and the experimental data is intriguing and suggests that maybe, I think what we need to do are longitudinal studies. You can't use brain striatum for that.